Afternoon, folks. All right, that's better. My name is Michael Ayers, and I serve as Dean of Math, Science, and Technologies here at Forsyth Tech. And it's my pleasure to welcome you all to our SciTech lecture series. We've given about 50 lecture series or lectures in this series, and had about 3,200 folks in attendance that were actually here physically. We have started broadcasting these live on YouTube, and so we may have people from around the country and perhaps even around the world watching our lecture series these days. So that is really cool. So I welcome each of you to the lecture hall today and hope that you get a lot out of our speaker. I know our executive director for the National Center for the Biotechnology Workforce, Mr. Russ Reed, knows these gentlemen very well, and he said he was going to introduce them, but he kind of snickered when he said that. It may be more of a roast than an introduction. What do you think? So Russ, come on up and introduce our speakers, and we'll go from there. Thank you all again for coming.
Well, good afternoon, everybody, and thank you, Michael. I really appreciate this relationship we have with your division, and really appreciate that. Greg Kucera, Dr. Greg Kucera, is a PhD. He's Associate Core Director of the Comprehensive Cancer Center here at Wake Forest. He's a professor of hematology and oncology, physiology and pharmacology, and cancer biology. He's a very talented guy. I happen to know Greg. I've known him for 16 years now, and I actually had the privilege of having him work with me with a company that we started with his dad back in 2001 called the Kucera Pharmaceutical Company. So when you see KPC-34, you're actually seeing a compound that was actually discovered through the process of having the company. But Greg is a graduate of Davidson College here in North Carolina. He's also did his PhD at Wake Forest, but he postdoced at the University of Vermont. Yay, UVM. Yeah, go for it. Yeah, and that's quite close to my original home, which is in Montreal. So played hockey against those guys. But he's had 25 years of research experience on novel therapeutics for the treatment of cancer. And he's been involved with research on 20 IRB-approved clinical trials at Wake Forest. And I think what's really interesting is that Greg has received numerous grants through the NIH and also through the North Carolina Biotech Center, and also has received, obviously through our company, some venture capital. But he's a pretty prolific guy. I went to his website just to check on how he was doing, and I guess publication means a lot, right, Greg? 57 publications and 32 since 2006. You know, you've got the resume, you can see, and I think that Greg's going to tell you that Tim Pardee was not able to show up today, but this has evolved to be a great partnership, which has taken a compound that literally was synthesized a decade ago and actually put it into action. And I think you're going to be very excited by what you see. So I'm very, very pleased you came today, and I hope that you'll take the time to ask Greg the questions that are in your mind, that you really want to you really want answers to as best as he can give them to you. And I want to tell you about the personal part of the Kuceras that I know. Greg's dad was a PhD and was at Wake Forest as well, and was a great inventor of very novel compounds for HIV. And that's how really I got to know him. Actually knew him back in the 80s by paper. And then I got the chance to meet with him and work with him in a company in 2001. Very tight family. And this is a family that really persists in academia. Greg's son is a medical doctor, and he's doing his residency now in surgery. He's in his third year. So these are guys that love medicine, obviously. So, but Greg, at the same time, and I don't want to make this too long, but I think it's important for you to know, he's very much a teacher, and he likes to interact with students and other faculty, and he's very, very highly respected at the Cancer Center and at Wake Forest. So without further ado, Greg, come on up and tell us what you know.
That'll be a short talk.
Thanks, Greg.
Is this on now? Did I turn it on the right way? Okay, good. Well, thank you very much, Russ. That was a very nice introduction. And yeah, my dad, I was with him this morning and told him that I was coming here, so he was all excited about that. Tim Pardee couldn't make it today. He had to go to the FDA, and then he's actually traveling to Israel after that. So he's a very busy guy. But this all would not have happened without the work of Tim Pardee and his animal model of acute leukemia. Also, Kucera Pharmaceutical Company, as Russ mentioned, they originally synthesized the compound that we're going to talk about today, KPC-34. And we're calling it a co-drug that combines a DNA damaging agent with a targeted therapy. Also like to mention that Pharmagra up in Brevard synthesized the batch of KPC-34 that we're using for these particular experiments. So here's an outline. I've got a number of slides because Tim was supposed to be here and I was supposed to give some and then he was going to give some. Well, I just merged them all together. So hopefully I can get through all of them. And if I start to see eyes glazing over with a lot of data slides, I'm going to just go really fast. And if you want me to slow down, raise your hand. Otherwise, we'll try and get through this. So I'm going to get a little bit of background about acute leukemias. I'm going to talk about the component of the KPC-34 that we've combined to a synthetic phospholipid, namely gemcitabine. Talk about the current therapy for acute leukemias, which has a backbone of cytarabine or Ara-C. We'll talk about how the drug bypasses several of the resistance mechanisms that leukemia cells utilize to bypass Ara-C therapy. I'll show you some of the in vivo experiments that, again, would not have been possible without Tim Pardee's laboratory's help, and then some summary and conclusions. So the acute myeloid leukemia, it's an abnormality of myeloblasts. So those are cells like neutrophils, eosinophils, and basophils. It's the most common leukemia in adults with about 18,000 new cases in 2014. And the mortality is fairly high at 10,000 deaths in 2014. It's a tough disease to have. And the prognosis is worse with the older you are. It tends to be a disease of the elderly. So 70% of the patients are greater than 60. Outcome is dismal. Oftentimes, the patients may go into remission, but the leukemia will come back. And the overall survival is only about 6.6%. The traditional treatment, as I mentioned earlier, is cytarabine. That's the backbone of the therapy. Notice here that that therapy was designed or first published in 1973, and they're still using it today. So there's definitely room for improvement. In this particular case study, there were 15 AML cases that were treated with this regimen that we call 7+3, which is 7 days of Ara-C treatment followed by 3 days of an anthracycline treatment. And Bob Capizzi, a hematologist, oncologist at Wake Forest, was one of the founders of this particular regimen called 7+3 that's in current use today. We get about 63% remission rate at first, but all of those patients are going to relapse with refractory AML. And that's known as the salvage regimen. So we're trying to salvage these patients whose leukemia has come back. And that uses a high dose of cytarabine. So we're using grams per meter square of cytarabine. We're using an anthracycline, mitoxantrone, and we're using asparaginase. And that helps to salvage some of the patients. But still, their survival rate is very abysmal. Another type of acute leukemia is acute lymphocytic leukemia. So this is a leukemia of the lymphoblasts. So those would be the B cells and the T cells. It's also called acute lymphoblastic leukemia. It's the most common cancer in children. So ALL affects children quite a bit. There are about 6,000 cases per year in the U.S. Fortunately for the children, it has a very high cure rate in children. So 80 to 90% of the children are cured of their acute lymphocytic leukemia. However, the cure rate in adults is only about 40%. So overall survival rate, we're looking at different age groups here. Remember I told you that acute leukemias tend to be a disease of the elderly. So if you're less than 30, your chances of overall survival are pretty good. So you get about 90% of the patients under 30 go into complete remission, about 47 are disease-free after 3 years, and their overall survival rate is about 58%. As you increase in age, though, these numbers drop. And so by the time you're greater than 60, only about 12% survive greater than 3 years. The treatment regimens for ALL are complex. The central drugs are anthracyclines, cyclophosphamide, steroids, vincristine, methotrexate, cytarabine, as we mentioned earlier. Routine CNS, or central nervous system prophylaxis, is important because this leukemia enters into the CNS and can cause paralysis. It can cause other deficits within the CNS, and so they have to use intrathecal injections of chemotherapy to try and prevent this from occurring. And maintenance therapy is needed for 18 to 24 months, following about 6 months of intensive IV therapy. So the principles of ALL therapy, you've got this induction. So you've got the patient, they've been identified that they have leukemia. They're treated with these drugs here, and that's known as the induction phase. You've got a consolidation intensification phase where you're using high-dose Ara-C, high-dose methotrexate. And then your goal is to just maintain these patients on a chemotherapy regimen of 6-mercaptopurine, methotrexate, vincristine, or steroids to try and prevent their ALL from recurring. And all along here, we're prophylactically treating their leukemia that may enter into the CNS. The overall survival, once they get relapse, again, is very poor. So again, we're looking at a disease that needs additional therapies for these patients that aren't doing very well. So here is cytarabine, or Ara-C. This is its structure over here. It's got a cytosine head group to this that's joined to an arabinose sugar ring, a 5-carbon sugar ring, and it's got a beta hydroxy position here. So it mimics deoxycytidine. And so we all know that deoxycytidine gets phosphorylated to its triphosphate form and then gets incorporated into DNA. The same thing happens with Ara-C. It gets phosphorylated to the triphosphate form. It enters into, is incorporated into DNA, and it blocks DNA polymerases. So the induction is complete but transient, so you get a remission of about 60 to 80%, and the relapse disease is very difficult to treat because the tumor cells become resistant to the Ara-C. Gemcitabine is another deoxycytidine analog, and the difference is in this, the 5-member carbon ring. Rather than having that beta hydroxy that makes it look like a deoxycytidine analog, it's got 2 fluorine atoms there. And it can also be incorporated into DNA and cause chain termination there. It's a newer drug than Ara-C, has been around. However, it has to be given IV, intravenously. The same thing with Ara-C and cytarabine. They have to be given intravenously. They're not orally bioavailable. Some of the benefits of gemcitabine over Ara-C is it more effectively inhibits DNA replication and repair. It also inhibits ribonucleotide reductase, and ribonucleotide reductase is an important enzyme for maintaining pool sizes of deoxynucleotides. When gemcitabine was first being developed, it was tested against Ara-C. It seemed to do much better in solid tumors than in liquid tumors like leukemia, but it was being tested in patients who had already failed Ara-C treatment. So, of course, the gemcitabine utilized the same activation enzymes and the same inactivation enzymes that would activate or inactivate Ara-C, so the gemcitabine just didn't work in leukemias. They had overlapping resistance. So this is how we see the cytidine analogs entering into the cell. They require an equilibrative nucleoside transporter, and that's shown here as this ENT. So the ARC has to utilize that transporter to get across that plasma membrane. It can't get into the cell unless it has that transporter. There's an enzyme, a rate-limiting enzyme, so without this enzyme, you won't get activation of ARC, that adds a phosphate, a monophosphate, onto the ribose ring. And then you've got other kinases that are going to add 2 more phosphates onto it to activate the ARC to its triphosphate form. Once it gets activated, it can be incorporated into DNA. The polymerases can add it to the growing DNA chain. Deoxycytidine triphosphate is a competitive inhibitor for that, so you want to lower levels of the competing nucleoside, deoxycytidine triphosphate. If you can do that, you can incorporate enough DNA or ARC into the DNA that can then block the DNA polymerases and cause apoptosis to occur, and the cells will die. Some of the limitations of the cytarabine-based treatments, you get poor pharmacokinetics, so the drug is metabolized very quickly. It's required to be given intravenously. It doesn't penetrate the blood-brain barrier very well, so we can't use this to treat any of the leukemia that gets into the CNS. It has a chemoresistance mechanism, so if you have a decrease in the equilibrative nucleoside transporter or an increase in the efflux pump, namely the multi-drug resistance pump, that's going to effectively lower the intracellular concentration of the drug in the tumor cells. If we don't have that rate-limiting enzyme, the deoxycytidine kinase, we won't be able to add that initial phosphate onto the nucleoside analog to activate it so that it can be incorporated into DNA. There are other deaminases that can inactivate the ARC to ARA-U, arauridine, that is inactive. In elevated PKC reduces apoptosis. I'm going to show you some data about why we want to target PKC. But elevated PKC seems to inhibit apoptosis and actually cause the leukemia cells to proliferate. And if you have an increase in cell cycle arrest, so the cells aren't going through the cell cycle, they're not going to make new DNA, so they're not going to incorporate the nucleoside analogs into the growing DNA chain, that can also reduce the effectiveness. And so these resistance mechanisms are correlated with a poor prognosis in the patients. So this is KPC-34. It's a gemcitabine monophosphate conjugated to aminophospholipid. So here we've already seen the structure of the gemcitabine. This is the phospholipid portion of the molecule. It's a synthetic phospholipid. Your body does not make a phospholipid like this. It's got an amide group here, and then it's got an ether bonded here. And we did structural activity relationships to come up with this specific compound that we call KPC-34. It's multi-drug MDR1 resistant. It bypasses the necessary need for that nucleoside transporter because it's made more hydrophobic by adding this phospholipid portion, so it can just melt through the plasma membrane, or it may use a receptor, a lipid receptor type mechanism to get across that plasma membrane, but it does not require that equilibrative nucleoside transporter. And once it's cleaved, it's only cleaved inside of the cell, inside of the tumor cells primarily, because they have mechanisms in place that can cleave this molecule. It's cleaved into 2 portions, and then one of the portions would be the gemcitabine monophosphate, and the other is a mimetic of diglyceride, which is actually a protein kinase C activator, but the mimetic here actually serves as an inhibitor of protein kinase C. So those are important things to think about as we go through the talk today. So this is how we envision KPC-34 getting across the membrane. There are some receptors, either through a lipoprotein receptor, we don't quite understand that, or it could just melt in like a steroid would get across a plasma membrane. But it does not require this equilibrative nucleoside transporter. So the resistant cells, even though they have lower levels of that, that's not going to affect the uptake of KPC-34. It's cleaved by what we think is a phospholipase C-like mechanism, and a phospholipase C cleaves phospholipids to the left of this phosphate, and it will leave that phosphate attached to the nucleoside and form the monophosphate form, and then the diglyceride is released. And that only happens in the cell because the tumor cells have elevated levels of the protein kinase C compared to normal cells. So this just shows that the diglyceride mimetic can inhibit protein kinase C, which is normally activated by diglyceride, and then we get apoptosis. So protein kinase C is the beginning of a proliferative pathway that signals through NF-kappa B, among other pathways, to cause the cell to grow and proliferate. Well, if we inhibit protein kinase C, we're going to inhibit the growth and proliferation of the cell, and the cell will undergo apoptosis. The gemcitabine monophosphate that we've formed as a result of that phospholipase C activity doesn't require that rate-limiting enzyme. We've already got the phosphate on there, so you don't need that deoxycytidine kinase to put that primary phosphate on there. And then we use these other kinases to phosphorylate it to the triphosphate form, and then it can go and act just like the gemcitabine triphosphate would do in incorporating into DNA and then blocking the DNA polymerases and causing apoptosis that way. So we're getting two mechanisms of action here. We're inhibiting the protein kinase C, and we're activating apoptosis. We're incorporating the nucleoside analog into the DNA. So now we'll look at some of the resistance mechanisms and how we determine that we're bypassing several of these resistance mechanisms. These are some brain tumor cells that we used, and we are using a drug called dipyridamole. Dipyridamole is an inhibitor of the equilibrative nucleoside transporter, and we're looking at gemcitabine versus the gemcitabine conjugate, KPC-34, plus and minus the presence or absence of dipyridamole. So if you've got dipyridamole in there, you're going to inhibit the equilibrative nucleoside transporter, and you're not going to get the nucleoside into the cell. And that's what we see here. Lower is better. So in this case, we're looking at an EC50, an effective concentration to kill off 50% of our cells. So we've got a low concentration of gemcitabine. That seems to do very well. But if we inhibit the transporter, we're raising that bar up, and it's taking more drug to kill the cells. Whereas here, when we're looking at the KPC-34, it doesn't matter if we have dipyridamole present or absent, because we don't require that transporter to get it across the membrane. So now what we're going to look at here, the effects of gemcitabine and the KPC-34 in cells that we've added additional deoxycytidine kinase to. So the rate-limiting enzyme, if you can think about it, if we add more of that rate-limiting enzyme, we're going to be able to activate the gemcitabine better, because it's going to add that first phosphate on. Whereas with the drug conjugate, because it's cleaved by phospholipase C, it already has that monophosphate, so you're not going to have any increased effect, and that's exactly what we see. So this is the IC50 value for gemcitabine in the cells that don't have an elevated amount of deoxycytidine kinase. When we add more deoxycytidine kinase to the cells, you can see very nicely that we see an enhancement in the effectiveness of the drug. Whereas these over here, where we're looking at the KPC-34 conjugate, adding increased amounts of the deoxycytidine kinase doesn't have a statistically significant increase in the activity of the drug. So that shows us then that deoxycytidine kinase is not necessary for the activation of the KPC-34. And then we looked at another resistance mechanism, the multi-drug resistant pump. So this is going to kick the drug out of the cell and reduce the intracellular concentration. And in this particular case, we're comparing it to doxorubicin, and we know that doxorubicin is a substrate for the multi-drug resistant pump. And you can see that when you have doxorubicin here by itself, this is the IC50 value. If you have elevated levels of that efflux pump, you would expect that you would need to add more doxorubicin to get the same cell killing, and that's exactly what we see here. So we have to add more doxorubicin to kill the same number of cells. Whereas with the KPC-34 conjugate, it doesn't matter if we have elevated efflux pump mechanisms there, because that the KPC-34 is not a substrate for that particular efflux pump. So here's some of the preclinical data that we have, and this happens to be on acute lymphocytic leukemia cell lines in culture. So we're just looking at the tissue culture assays here. We use a 96-well plate mechanism. It's a colorimetric assay. We'll plate the cells, and then we'll treat for a couple of days, and then we'll determine the cell viability by the color change that we see. And these are just a number of different cell lines that we have in the lab. These are all acute lymphocytic leukemia cells, and these are acute myelocytic leukemia cells. They have different properties, but you can see that the IC50 values against all of these different cells, regardless of their different properties, their P53 status, FLT3 ITD status, they all have IC50 values in the nanomolar range, which is a very good level for us to be working in. If we look at a measure of apoptosis in the presence of KPC-34, we're using a flow cytometric assay here. So we're treating the cells and we're labeling them with either annexin 5, and that's going to bind to phosphatidylserine that's on the external part of a cell as it begins to undergo apoptosis. So the more annexin 5 binding you have, the more apoptotic cells you're going to have. And we're also using a dye called propyridium iodide, which binds to DNA, but it can only bind to the DNA when the cell membrane has been fractured. And so ideally what you want to see is a membrane that's intact, so low PI levels, but high annexin 5 levels, and that'll show you the amount of apoptosis that's undergoing. That's taking place. And so these are the non-treated cells here. So there's very little, and this quadrant here would be the apoptotic fraction of the cells. And you can see as you add increasing amounts of KPC-34, you get increasing amounts of cells that light up in that apoptotic quadrant. If you have any questions about any of these assays, feel free to raise your hand and ask, so I'll explain it a little better. So this is just a graphic representation of the data that you saw on the previous slide. So we'll see a reduction in the cell viability of those cells, as well as that flow cytometric assay. The other thing we were interested in looking at was, does this KPC-34 affect the protein kinase C activity of the cell? Protein kinase C depends upon phosphorylation in order for it to be activated. And so what we're looking at here, this is a Western blot of phosphorylated protein kinase C. So we're using antibodies to look at the levels of phosphorylated protein kinase C. And then we're comparing that to a standard in the cell gamish that we know should be fairly constant over the different treatments, and that's actin. So after 4 hours of no treatment, we see a very nice phosphorylated protein kinase C. And then as we increase the time with a constant dose of KPC-34, you can see that you see a reduction in the amount of activation of protein kinase C as shown by the decrease in the phosphorylation of protein kinase C. And that's just shown graphically over here. So if we look at some ALL cells then, and the downstream signaling that takes place, PKC is the beginning of that proliferative pathway, and some of the downstream signaling that takes place when you're causing these cells to undergo proliferation is the NF-kappa B. And NF-kappa B is another signaling molecule that's going to, it's a transcription factor that's going to turn on genes and cause cells to grow and proliferate. But you can see very nicely that we're inhibiting the phosphorylation of NF-kappa B by treating these cells with KPC-34. So we're not only inhibiting the phosphorylation of protein kinase C, but we're inhibiting the downstream phosphorylation of other effector molecules that would cause the cells to undergo growth and proliferation. So now we'll get into some of the in vivo experiments that Tim had done. We did most of these in vitro assays in my laboratory. I had very little experience with working with animals, and it wasn't until Tim joined our faculty probably 5 or 7 years ago, and he brought with him this mouse model of acute leukemias. And we got together, and I said, Tim, let's try it and give it a shot. So here's what he would do. He would take his B6 acute leukemia cells, and he engineered luciferase into those cells. And luciferase is the same thing that you see in a firefly. So at night during the summer when you see the firefly light up, that's exactly what we can do when we add ATP and luciferin to the cells. We can see these cells light up wherever they are. So we take these mice and we inject the tumor cells into them. They engraft the leukemia very nicely. And then we treat them either with gemcitabine or KPC-34 or just saline as a control. We have to use gemcitabine intraperitoneally or IV because we can't give it orally, whereas with the KPC-34, we can give that orally. And I'll show you some experiments where we treated the animals intraperitoneally and IV with KPC-34 as well, and it doesn't work as well. And I'll give you an explanation of that a little bit later on. But nevertheless, we would treat the animals, and then we would image them 24 hours later to look for the luciferase in there, the lighting up of those tumor cells, and we would also measure their survival and see how long they would survive. So these are anesthetized mice, and we're just using a camera that can measure the amount of luciferase there, the amount of luminescence that's present. And when these 3 mice here were treated with KPC-34, and these mice over here were treated with gemcitabine. Well, these guys look pretty good. There's not much luciferase lighting up in there. But and this is 10 days post-treatment, but if we look at their survival curve, so we're looking at these animals, how long they will survive. And our survival, we didn't let them go until they would die. Our animal protocol was very specific about that. As soon as they begin to get the hind limb paralysis, we would sacrifice the animals. So just to protect them just from suffering too much. And so you can see that with gemcitabine, their survival was about approximately the same as the control cells. Whereas with KPC-34, look how much longer these animals would last before they would succumb to the hind limb paralysis. And down here, we're looking at the median survival time. So with gemcitabine, we're at 15.5 days versus almost a doubling for KPC-34. So this looked really good. We were very excited by that. So we began to explore more, and we looked at, is it superior to gemcitabine? So we took these B6 ALL cells, injected them into the mice. We treated them with gemcitabine, KPC-34, and saline, or either IP or IP or orally, and then we had the idea that we were covering up some of the luminescence from their skin. So we would sacrifice these mice and very quickly then look at their tissues underneath the skin, and so we can see that with the KPC-34 mice, this is again what we saw in the previous slide where we were looking at the mice that were just anesthetized. These mice have been sacrificed, and now we're looking at the bioluminescence in the mice. And it clearly shows that there is no bioluminescence with the KPC-34, in particularly in the craniums of these animals. And down here you can see the gemcitabine mice. So even though we didn't see it in the mice that still had their fur on them, when we took the fur off, we could see that they still had tumor present in the animal. So we're looking at then KPC-34 and how it can reduce the leukemia that gets into the central nervous system. So we just quantified the amount of bioluminescence, and that's shown here. So we took the data from the previous slide, and we're looking at, this is just the saline control here. This is in the whole body and then in the brain, and you can see that there's a nice reduction in the amount of luminescence, especially with KPC-34. And if we look histologically at the brains of these animals and we compare gemcitabine to KPC-34, this is what leukemia looks like when it crosses the blood-brain barrier and gets into the CNS. It stains this dark blue color. And this is just another control-treated animal. With gemcitabine, you can still see that there are leukemia cells present here, whereas with the KPC-34 animals, there's virtually no tumor cells present in these histology slides. So this is looking at a human acute lymphocytic leukemia cell line. So as Russ mentioned, I'm one of the associate directors for the Tumor Tissue Pathology Shared Resource of our Cancer Center. So we're able to have access to leukemia cells from patients, and that's what we're looking at here. We're looking at, this is an in vitro experiment, so we've just taken the leukemia cells out of a patient. We're treating them either with saline control or with KPC-34, and the leukemia cells that come from the leukemia patients are still susceptible to KPC-34 treatment. The standard of care for acute leukemias is not just Ara-C. Remember I mentioned that they also utilize an anthracycline, and doxorubicin is the anthracycline that we're using here because mice cannot use adriamycin, and adriamycin is the usual anthracycline that humans use. So we incorporated doxorubicin, which is similar to the anthracycline that's used in humans. But again, we treated mice with the leukemia cells. We allowed them to engraft, and then we would treat with either Ara-C plus doxorubicin or KPC-34 plus doxorubicin or saline control. And then we look at the bioluminescence of the mice using the IVUS. And so this is the Ara-C doxorubicin. Here's the saline control, and then KPC-34 plus doxorubicin. And then if we look at the survival of those animals, you can very clearly see that the KPC-34 plus the doxorubicin, so this would be considered a comparison in a mouse model of the standard care therapy that humans would receive. And so this is the Ara-C plus the doxorubicin compared to the KPC-34 plus doxorubicin. And again, we've increased the survival of these mice from 30 days out to about 50 days. So we saw the in vitro experiment where we added dipyridamole to the cells to block that equilibrated nucleoside transporter, and that showed that we didn't need that for KPC-34. So one of Tim's postdoc, our graduate students, and then later a postdoc, Pete Alexander, had the idea, let's try and treat the mouse with dipyridamole and see if we can affect the efficacy of Ara-C versus KPC-34. I really didn't think this experiment would work, but Pete proved me wrong. So he took the animals, he got them engrafted, and then he would pre-treat with the vehicle, or he would pre-treat with dipyridamole, and then he would treat with either the saline control, the gemcitabine, or KPC-34. And this is what we see here. So this is just the vehicle treatment here, and then this is vehicle plus gemcitabine, and then we've got the dipyridamole plus gemcitabine shown here, and the dipyridamole actually inhibited the gemcitabine experiment, just as we thought before. Whereas with the KPC-34, it actually helped the activity. And so if you look over here, looking at the saline plus dipyridamole and saline plus vehicle, it's 13 days. If you look at gemcitabine plus the dipyridamole, it inhibited it somewhat, but it was statistically significant. Whereas with KPC-34, it actually augmented the survival from 20 to 28 days. So remember I told you that the mice would get hind limb paralysis and then we would have to sacrifice the mice? We said, well, let's try and retreat the mice with KPC-34 and see what happens then. So we took the mice, we got the leukemia engrafted in the mouse, and then we took the Ara-C and the anthracycline, we treated the mouse, and then we waited for hind limb paralysis, and then we retreated the mouse up to 2 times with KPC-34 orally to see what would happen. And so this is a picture or a movie of what we saw when we did that experiment. So this is going to show a mouse that was treated with Ara-C and the anthracycline, and you can see that the hind limbs are splayed out. The mouse can't move them very well, and within 12 to 24 hours, this mouse would die if we didn't do any other treatment to it. So we were just sacrificing the mice at this particular point. And then we treated a mouse with KPC-34, and 24 hours later, this is what we see. The legs are up underneath the mouse. It's moving around a little bit better. Seems to have a little more activity, but it's still not 100% yet. It still has a little bit of that hind limb paralysis taking place. And then this is 48 hours after an additional treatment. And you can see the mouse is almost back to normal. The legs are up underneath. It's not exhibiting hardly any hind limb paralysis at all. So that's just after 48 hours and 2 doses of the KPC-34. And now, if we take that mouse, it might begin to get hind limb paralysis again, but we can retreat again with additional KPC-34, and it doesn't become resistant to the KPC-34. So this is what we see with just a saline control mouse. This is just the historic data here. But if we took the mouse and if we began to see hind limb paralysis, and we retreated these mice, we could extend their life even greater by just one additional KPC-34 treatment once their hind limb paralysis occurred. So if we retreat mice with KPC-34, this is a representation of the experiment that we're showing here. Let's treat them multiple times with KPC-34. How long can we keep these mice alive? And that's shown in this particular slide. So we can retreat these mice on a weekly basis with KPC-34, and look how long we can extend their life. We're on out, in one particular mouse, we were past 200 days treating this mouse. So he was effectively cured of his leukemia. So now what happens if we take KPC-34 and observe resistance in an ALL in vivo model? So what we do here is we graft the leukemia cells into the mouse. We treat with Ara-C to get resistant cells. And then we harvest those cells from the mouse and take those Ara-C resistant cells, reintroduce those into a new mouse. So the leukemia takes hold in those mice, and you would expect that the leukemia in these mice would be resistant to Ara-C. So we're treating here with Ara-C. We're also treating with gemcitabine and KPC-34, both intraperitoneally and orally. Remember, I told you that we get better activity when we treat orally for some reason than we do with intraperitoneal injection. And so these would be the data to support that, and we're comparing that to saline. And this just is the IVUS representation of that. And then if we look at the oral administration of KPC-34, reducing the leukemic burden compared to Ara-C and gemcitabine in these cells, this is the graph of that slide that we just saw before. So this is Ara-C, virtually no activity against the bioluminescence. Gemcitabine had some activity, whereas KPC-34 was the best. And if we compare the oral dose versus the IP dose, remember I said that the oral dose was much better. You can see that in this particular slide. So now if we look at the survival of those mice, again, these should be resistant to Ara-C and to gemcitabine, and that's exactly what we see. So here are the mice that have the Ara-C. Here are the mice with the gemcitabine. And then these are the mice with KPC-34 given intraperitoneally. So it's virtually about the same as the gemcitabine. whereas this curve out here shows the mice that had the Ara-C resistant cells that were treated with KPC-34. So those mice lived out to about 24 days, even though they were resistant to Ara-C and to gemcitabine. So there's no cross-resistance there. So let's look at the effect of oral dosing versus intraperitoneal dosing of KPC-34 on DNA damage induction. So we're looking at the ability of the drug to break the DNA, to cause chain termination of the DNA. And that's what we're going to do in this particular experiment. So we're using the oral dosing versus the intraperitoneal dosing. And again, what we're looking for are cells in this apoptotic quadrant over here. H2AX is another marker that we used for apoptosis. It would be similar to the annexin 5 that we used in a previous flow cytometric assay. But what you're looking for is an increase in the number of cells that appear in that lower right quadrant. And you can see very nicely that we get a nice response when we give the drug orally, but we see less of a response when we give the drug intraperitoneally. And then this is just a graphical representation of what we showed in the previous slide. So I told you that orally is better than intraperitoneally. So you would expect that this phospholipid conjugate of gemcitabine had to be taken up in the small intestine, and it was probably taken up in a way that fats that you eat in your diet are taken up. And so we wanted to test that in this particular experiment. So we were looking at the effect of dietary fat on the uptake of KPC-34. And we think that the KPC-34 would be taken up into by the small intestine and packaged it into chylomicrons, which is a way for transporting lipids into the circulatory system. We wanted to see if a high-fat or a low-fat diet would affect that. And you would expect that if you had a high-fat diet, you would inhibit the efficacy of KPC-34. So that's what we're testing here. So for looking at the effect, so this is a low-fat diet with KPC-34 versus a high-fat diet, KPC-34. This is just the bioluminescent scan. But if we look at the survival of these animals, you can see way out here that we're looking at the low-fat diet with KPC-34 is much better than when we have a high-fat diet and are using KPC-34. So we can expect then that if we are going to ultimately use a clinical trial, we want to make sure that the individuals that are taking this particular drug are on a low-fat diet so that we can get more uptake of the KPC-34. Now we're going to go to AML. So that was acute lymphocytic leukemia, and we've got a few minutes left. So we began with the acute lymphocytic leukemia because the cells responded very well to that. We knew that it depended upon protein kinase C to get that optimum response. And Tim was looking at acute myelogenous leukemia cells from patients and using KPC-34 in an in vitro assay, and he just wasn't getting much luck there. And so he decided to look at the protein kinase C levels in the cell, and that gave him a clue that some of these different patient cell lines, so these would be the different patient cell lines, had different levels of activated protein kinase C, and that's exactly what we saw here. If we look at protein kinase C alpha, so this is a Western blot just of the amount of PKC protein. We're not looking at activated protein, namely the phosphorylated form, but you can see that the different patient cells have different amounts of protein kinase C enzyme protein in them. And the same with the beta-2 isoform of protein kinase C. There are several different isoforms, but we think that it's these two isoforms that are important. So this could potentially explain why some of the AML cells responded to KPC-34 and some did not. So Tim went back and looked at some of his AML cell lines, and he found the same thing. So he had these different cell lines, the HL60s, the KG1As, and so on and so forth. But it wasn't the only ones that expressed phosphorylated or activated PKC were the HL60s and these OCI AML3 cells. So he used those in some subsequent experiments, and he was able to show that increasing amounts of KPC-34 could inhibit the activation of protein kinase C. That's shown in this particular Western blot. So he took those particular cells, or he used these cells here, the MFL2s, which are back here. They did not have protein kinase C, activated protein kinase C. So he introduced those cells into the mouse and he treated with KPC-34 or saline, and you can see that there's no difference in the activity. So this begins to give us a hint that we need that protein kinase C in there for KPC-34 to be better than the other nucleoside analogs that are out there. And then he took those ones, the OCI cells that had protein kinase C in them, did the same experiment with KPC-34 and saline, and look at this. You can see a very nice increase in the survival of the mice that have that protein kinase C and that can be activated. And so we're looking here at retreated mice. So again, these are the same mice with that OCI AML cells. These are just treated once with KPC-34, but if we retreat those animals when they begin to get that hind limb paralysis, we can go from 22 days to 44 days on out to 78 days with that retreatment. Here we're comparing those OCI AML cells to ARA-C, and you can see that ARA-C virtually is ineffective at treating those OCI AML cells that are introduced into the mouse, whereas KPC-34 is very effective. So we go from 25 days of survival out to about 44 days. And then Tim was able to generate a PDX model, or patient-derived xenograft model of AML. So he took AML cells from a patient. He was able to grow those in an NSG mouse and get the leukemia to engraft into those mice. He treated with either KPC-34 or saline. And he tested a number of different AML cell lines cells from these patients and tested them for total and phosphoprotein kinase C. And you can see that some of the patients had very nice levels of phosphorylated PKC versus some that didn't have very good levels. And in the PDX model that had high levels of protein kinase C that could be phosphorylated, you can see that these are the leukemia cells that are growing in the liver and in the spleen. They would be the brown colored cells, whereas animals these are two different animals that are treated with KPC-34. You can see a nice reduction in the brown coloring, indicating that the KPC-34 is effectively inhibiting the growth of those leukemia cells in the mice. These cells were not labeled with luciferase, so we couldn't use the bioluminescent assay. So the only way we could check for the leukemia was to sacrifice the mice and then look in their liver and spleen. It's well tolerated in vivo, and that's shown in this particular experiment. So we've treated with KPC-34 and gemcitabine, and we're looking at different tissue types. So this is bone marrow, spleen, and liver, and they virtually look the same. So KPC-34 doesn't seem to be affecting either the bone marrow, the spleen, or the liver in a negative manner. These are mice that that don't have leukemia. So we're just treating the mice with the drugs and then harvesting sacrificing them and harvesting their organs and looking for any damage. And again, we don't see any damage with KPC-34 in small intestine, large intestine, kidney, or pancreas. So here we're looking at, this is my final data slide here, and Tim just generated these data very recently. So he took this mouse, this PDX mouse that was growing this AML 210. So this was an AML cell line from patient 210. He was growing the leukemia in those mice, and then he would retreat with KPC-34 over time. And these are just 3 different mice shown here. This is a control mouse that wasn't treated at all, and we're looking at phosphorylated protein kinase C. And what he found out was the mice that became resistant to KPC-34, they were losing their activated phosphorylated protein kinase C. So this again adds evidence to the idea that we require protein kinase C in the leukemia patients in order for the drug to be effective. So summary. Overall stromal chemoresistance in vitro, I didn't show you any data in that, but in leukemia, the stromal cells can actually provide resistance against Ara-C. It inhibits protein kinase C phosphorylation in both ALL and AML. It's active against ALL and AML in vivo. Superior to gemcitabine in ALL, it reduces CNS burden in ALL. It's more effective than Ara-C combined with doxorubicin. It's tolerable as a salvage agent in relapse standard care ALL. It's active against CNS disease, active against Ara-C resistant ALL. There's a benefit with the oral delivery versus IP delivery, and it's well tolerated. And so KPC-34 is a novel drug against multiple models of naive and chemoresistant leukemia. Two distinct anti-neoplastic mechanisms, namely targeting the DNA and protein kinase C. Oral bioavailability approves efficacy. It dissolves very well in water, so we can just mix it up in water and give it to these animals. It has the potential to replace Ara-C therapy either as a single agent or combination in naive patients. We could use it as a single agent in relapse refractory acute leukemias. And it also could be used as a CNS prophylaxis in those acute lymphocytic leukemia patients. We are still playing with the dose and the schedule, how to optimize that and reduce toxicities, and it further justifies transition into the clinic and development of a companion diagnostic, namely, can we identify the patients that are going to respond to the KPC-34 by having a diagnostic test that we can use. These are the people that did the work. Pete Alexander was the graduate student, later a postdoc in Tim's lab, of course, Tim Pardee. Rich Alexander was a graduate student in my lab who synthesized some earlier Ara-C gemcitabine phospholipid conjugates, along with Carrie Pick-and-Palmy, who was a postdoc in the lab, my longtime technician, Gilda Saluda. These are other folks in the Pardee lab. Folks I've left off of here were Jan Hess and Russ Reed and Ron Fleming at Kucera Pharmaceutical, as well as Khalid Ishak and Susan Morris-Natchke at UNC, our longtime collaborators. And this was funded by an NIH training grant. Wake Forest Innovations has been a very strong supporter of developing this technology, and without them, we couldn't have gotten as far as we have. And then these are some other foundations that have supported our research. So I'd be happy to take any questions, and I hope you were able to follow along.
I just wanted to find out what the time frame would be for it to get to clinical trials.
It's not the time frame, it's the cash involved, I think, is more critical. What we need to do now are to do IND-enabling toxicology studies in a non-rodent model, and in order to do that, we would need sufficient amount of drug to do those studies. We have that lined up through folks that have made the compound for us before. We're just looking for additional funding now. We've got support of our cancer centers to do a quick clinical trial, which could be done in a couple of years at the most. So we see a large number of acute leukemia patients at Wake Forest, so the patient population is there. We also have the refractory patients that could benefit from this treatment. But that's a good question. I think we could do it in a couple of years.
I guess recently the CRISPR, the gene editing technology, seems to be kind of in the news. Would this benefit at all by getting the drug specifically into the cell?
So I could see an advantage there in patients that didn't have the protein kinase C in their leukemia cells. I'm not as familiar with the CRISPR technology to say that we could target it specifically to leukemia cells versus normal cells. The advantage of this technology is that you're bypassing a number of the resistance mechanisms that CRISPR could eventually fix, but we don't have to worry about it. We're able to bypass those irregardless of the CRISPR technology.
Okay. My other question was just in regards to side effect profiles. I mean, what
so
have you heard?
the side effects that we see are very minimal. I showed you those histology slides, so it doesn't really affect the histology of a lot of things. If we go too high in concentration, we typically use about 20 milligrams per kilogram of mouse. And if we go above that, we begin to see toxicities, namely the mouse just doesn't look right. They get what we call a piloerection. Their hair begins to stand up on end. They just don't act right, and some may even die early before the drug can have an effect. So we figured out that a lower dose is much better, but you have to have an optimum dose there. That's why I mentioned that dosing and scheduling is very important.
Okay.
Yeah.
Thank you.
Any other questions?
Yeah, you'll need it for them to hear you out in the cyberspace.
All right, so then I can tell a joke.
Okay.
That was very nicely presented, very nicely controlled. Surprising, one of the most surprising things is the improved efficacy of the oral versus interperitoneal, and I'm curious as to whether you might imagine the possibility that there are some signaling pathways, even from the satiety centers or olfactory centers or gut, et cetera, all the way down through the stomach, and what might happen, for example, if you administered directly to the stomach.
Yeah, that would be a good experiment to do. Somebody asked me about using an infusion pump, pumping it directly into the stomach, and I sort of, and the reason they wanted to do it because they thought that gavaging the animals was too difficult. But I don't know if that would be a good experiment to do, but I just don't know if there are things in the alimentary canal above the small intestine that could affect the uptake of the drug. We haven't done any experiments on this, and what I would like to look at are the chylomicrons. So that's where fat is initially absorbed from the small intestines. It's taken into the lymphatic system and then into circulation. Those chylomicrons are taken up by the liver and repackaged into very low density lipoproteins, and then those are secreted out of the liver and go to extrahepatic tissue, and they're acted upon by lipoprotein lipase and other enzymes that take the lipid into the cell that is utilizing that lipid as an energy source. So it could be that it's that lipoprotein particle that the cell sees and says, wow, I need additional energy because I'm growing very rapidly. I want that fatty acid and those phospholipids to maintain my plasma membranes and to maintain my energy source. So that's why I'm taking up a lot of fat from the very low density lipoproteins. It could also just be that the drug is in single suspension in the circulation, but we don't have any evidence for that. We need to look. Uh-huh.
Simply wondering whether there may be a mechanism that's independent of the lipid metabolism
Yeah,
and digestion.
yeah, that's a good question. I have no idea. If somebody has the smarts to figure out an experiment, we could try it. Yeah.
Anyone else?
Hi. So why is the cure rate so much more successful in children as opposed to adults that have leukemia?
Yeah, so that's interesting because I think there are a couple of different reasons there. One is when you have an older individual, you tend to think that they're more frail and you don't want to treat them with the bigger guns in chemotherapy, so you may not give them the maximum tolerated dose that they could actually take. Versus a young child, they're very resistant or resilient, and they don't seem to have the side effects that an older individual has to the chemotherapy regimen. So it's a combination of treating the older patients with less chemotherapy than the younger individuals, and it's more that the younger individuals, their vitality is greater, and they're able to respond to the chemotherapy without the side effects. That would be a better question for Tim than me. I don't think I'm giving you a satisfactory answer there. But
I have another question. So the drug only causes apoptosis in the cancer cells, correct?
well, yes, that's the mechanism that we're mainly looking at. Yeah.
Thank you.
Anybody else?
So what percentage of the apoptosis do you think is coming from the PKC pathway versus the DNA incorporation pathway?
Yeah, I don't know. I got to stand behind this flag. We never really quantified that, so I couldn't give you a good answer to that. But if you looked at the cells that didn't have protein kinase C, they were less affected by the KPC-34. So I guess you could look at the difference between those that had a full complement of the protein kinase C versus those that were deficient in protein kinase C, and that would be your amount of difference that you're looking for. But we never really did those experiments.
So
Or
if
quantified them.
if we would say that it's possibly working quite a bit through the PKC pathway, would there be any benefit to giving this drug in addition to the gemcitabine, but you're essentially doubling up on the DNA incorporation pathway?
Yeah, we never tried that. That would be interesting to look at. Yeah, good idea.
So, Greg, should we have gone cancer or should we have gone HIV in 2001?
Hindsight is 20/20, right, Russ? Yeah, I don't know.
You don't have to answer.
No. So one observation I think that is very important here is that you need to have the right readout for what you want to measure. You can't depend upon historical readouts in order to get an answer. And as we found out here, oral was better than intraperitoneal. So until we did the experiment, we didn't know. You have to do the experiment, and you have to do an experiment with the appropriate controls in order to validate what you're trying to show.
So I'm wondering if you remember when we made the presentation down at RTP to the investors, and we told them we thought it was orally bioavailable, and they all looked at us and said, well, that's not an advantage. And the cancer experts said, no, that's how we make our money.
That's exactly right. So a lot of revenue is generated in oncology by giving drugs in the clinic. However, we're always looking for improving the quality of life in these patients too. That's just as much of a factor. In fact, gemcitabine was approved in pancreatic cancer, not on its efficacy at treating and improving the overall survival, but it improved the quality of life of the individuals. So that's why gemcitabine now is the standard of care for pancreatic cancer.
Given that anthracyclines are cytotoxic and have very bad side effects for multiple organs, is there any thought of doing a dose response with lowering the level of anthracyclines given as co-treatment with your drug?
That again gets to my story of dose and scheduling. We really haven't played a lot with the anthracycline doses, but you're right, they're very toxic. The other thing we saw was, remember there was the experiment that we looked at multi-drug resistant efflux of the anthracycline, and when we put gemcitabine and the anthracycline together, the KPC-34, it seems to actually inhibit the multi-drug resistant pump. So that could actually enhance the activity of the anthracycline and allow us to reduce the amount of a therapeutic dose for the anthracycline.
Anybody else? Dr. Burrell?
Dr. Kucera, I just would like to say thank you for taking the time out to share your work with us, for enlightening us about the KPC-34 drug and its promise in cancer therapy. And I wish you success as you continue your work.
Thank you very much.
Hopefully you'll be able to help a lot of people.
Yeah. Thank you.
Thank you.
Yeah. Thanks for coming.
So Greg, we know you had to work hard, so we wanted to make sure you got another diploma. So here you go. We have one for you and one for Tim. Thanks very much. Here you are.
Thanks. Very nice.
There you go. Yeah, we're gonna take pictures.
Yeah, okay.
And there's one for Tim. Great. So we'll put that down. Take some pictures. Yeah, come on, Cheryl. We'll take some pictures. Thanks. Thanks, man. Thanks, man.